So to have this disclosure slide, my research office requires that, so there it is. The setting for the talk, and I think for a lot of the workshop, is tumor evolution and the fact that tumors evolve over time. They accumulate somatic mutations, both passenger mutations and consequential driver mutations. The end result being when we observe or sequence a tumor at the present day, we have this heterogeneous mixture of cells as a result of this accumulation of somatic mutations and subsequent clonal expansions. So the goal in a lot of the work that probably you'll hear about over the next couple of days is to really infer this process of tumor evolution from DNA sequencing data. And in most of my talk, I'll actually be talking about bulk DNA sequencing data as opposed to single cell sequencing data, because a lot of the data we have is from bulk sequencing of mixture. So how can we start to quantify and infer the past history of this process? And this is, as you all know, and I know David gave a tutorial yesterday on phylogenetics. This is a process that, like any evolutionary process, can be quantified in terms of a phylogenetic tree. So what we can attempt to do is take one or more parts of our tumor, sequence them, identify all the somatic mutations in those pieces, and then use various existing tree reconstruction algorithms to build this tumor phylogeny, here represented by a tree where each node is a clone or group of cells that contain the same somatic mutations, and on the edges are labeled by the mutations that have accumulated from the time from the parent to the descendant. Now, there's many reasons we might like to infer these trees, and there's many applications of these trees, really understanding what is the mixture of cells, trying to quantify things like drug resistance, et cetera. But the one I'll talk about today is using evolutionary trees to try to understand something about the process of metastasis. And metastasis is when a cell leaves the primary tumor and seeds a new tumor at another anatomical site. And it's important to understand this process because metastasis is really the big killer in cancer. Most patients die as a result of metastasis and not as a result of the primary tumor. So historically, the process of metastasis is thought of as metastasis being a monoclonal event, which is the result of a single cell migrating through the broader lymphatic system and seeding a new tumor at a distant site, and then that tumor will grow out and accumulate new somatic mutations. And then perhaps later a cell leaves of a primary tumor, or perhaps another metastasis seeds a new tumor at another site. And so this monoclonal theory of cancer has been sort of the dominant paradigm for how we think about metastasis occurring. But now we could actually imagine taking sequencing data from the same patient, from the primary tumor, and from one or more metastases from that patient, and then using phylogenetic techniques to attempt to reconstruct the past. And in this case, not only are we interested in reconstructing the tree itself from the mutations, but also reconstructing, if we can, where and when these cell migrations occurred. And then perhaps we might see that there may be more migrations between these different metastases. And in fact, I'll show you some data that suggests that there can be. So this has been a very popular topic in the past few years because of the availability of sequencing data from primary tumors and metastases from the same patient. And here's just a few of probably now dozens of examples that have been published. And on the left here is a colorectal cancer. Again, single patient. This tree reconstructed from 5 sections of the primary tumor and 2 liver metastases. An example from a mouse skin cancer. Here there were 2 parallel carcinomas that were in this mouse model of skin cancer and multiple Mets. And by building the tree, you can see some Mets seemingly are more closely related to one carcinoma than the other. Another example in breast cancer, each tree again from a different patient, multiple patients sequenced, and you can see the metastases and their relationships to the primary from each patient, and the mutations that were shared amongst the primary Mets and the ones that were newly acquired. And we can start to learn a lot about this process by building such trees and sort of seeing, in this case, that the 2 Mets are more closely related to the primaries. So we started to dig into this a little bit, and just to sort of zoom in on the one tree I showed you, here is now this tree. It was, again, 5 samples from the primary, 2 liver Mets, and there were 412 single nucleotide mutations that were used to reconstruct this tree. And when we went and took this tree and layered the mutations back on top of the tree, each edge labeled by a certain number of mutations, what we saw that was about 10% of the mutations occurred more than once on the tree. And this is a very interesting evolutionary event, the so-called homoplasia event. So you've got a single nucleotide mutation that's happening independently twice during the evolution of the tree. And the edges here are labeled by the number of such homoplasia events. So for example, looking at this edge here, there are 20 of the mutations on this branch are elsewhere on the tree. On this branch, 24 of the mutations are elsewhere on the tree. In fact, most of these 20 and 24 are actually the same mutations. So somehow what seemingly happened, if you believe this tree, is that mutations that distinguish the two metastases in this section of the primary occurred on this branch, and then independently, at another time, many of those same mutations happened in a different part of the primary tumor. This is a quite amazing event, and this is quite a high level of homoplasia. But it's really kind of a curious result. I mean, often this homoplasia, so-called convergent evolution, is really a very interesting event evolutionarily, and to see such high rates of homoplasia is really quite curious. And so one could perhaps say, well, maybe the way of building the tree was not correct somehow, and in this case they used maximum parsimony, and maybe they should have used a Bayesian approach, et cetera. But it turns out that that's not really the reason for why there's this curious result. Instead, the reason why there's this curious result is that each of the samples that were sequenced, although they were multiple samples from a tumor, each of them is not a homogeneous population of cells. It's a heterogeneous population with different somatic mutations. So by pretending that each sample was a collection of cells with the same mutations, we get this very strange apparent homoplasia. And this is sort of an example of how sometimes we have to be careful in using existing algorithms in analyzing new types of data. And I don't know if David showed this to you yesterday, but he sort of wrote a really nice position piece reminding the cancer biologist that when you construct trees from different regions of the tumor, this is not the same as building genuine phylogenies because of this, this heterogeneous mixture of cells. So how then can we start to get a better handle on this picture? So if we want to understand metastases, if we take our samples and we build a matrix that tells us it's a classic sort of phylogenetic matrix, whether a mutation is present or not in each sample, and then we try to build the tree, then underlying that is this assumption that each sample is homogeneous. And if we make that assumption correctly, we might get these strange results like this apparent homoplasia. So that's not really the way to address the issue from data like this. What we really need to be aware of is we need to build trees that are not on samples, but build trees on individual cells or on clones. And so there's a whole other line of work over the past few years to try to build phylogenetic trees from these mixed samples. And these are the so-called clone trees that I was describing, where now each sample has been split up into multiple clones that each share the same somatic mutations. So it turns out doing this is not trivial at all. It's in fact quite complicated to build a phylogenetic tree from mixed samples. And there's different issues that arise if you're looking at different types of data, if you're looking at single nucleotide mutations, or if you're looking at copy number aberrations, if you're trying to use both. It gets quite challenging quite quickly. And just to show you sort of some of those issues in brief, and I'm sure you'll hear a lot more about them in some of the other talks. If we say look at perhaps single nucleotide mutations, and we think about 3 different samples of the tumor, and then if we try to represent these samples according to the presence or absence of different somatic mutations that we've identified, this would be like the classic input for a phylogenetic approach, but that's not really what the data tells us. In fact, in each sample we know not just about the presence or absence of mutation, but if we go think about how these mutations were detected, they're detected from the alignment of sequence reads to the reference genome. And so really when we look at the underlying sequence reads, we get some additional information because we also obtain some information about the number of such reads that cover that position that have the mutation. And that number or fraction of such reads is the so-called variant allele frequency. So this variant allele frequency, the fraction of reads covering a position that have the mutation, is actually proportional to the fraction of cells in that sample that have the mutation. So what's happened is that this binary matrix, if you will, is digital data has become analog data. because now we have these frequencies that come from somehow the fraction of reads that cover the genome. So now the phylogenetic problem that we need to solve is not one of, given this presence-absence data of mutations, build a tree, but instead we have information not about the leaves of the unknown trees. This is the classic phylogenetic problem. Given the mutations at the leaves of the tree build the tree. Instead, we have a problem where we're given the mixtures of the leaves of a phylogenetic tree, and we have to uncover that tree. So, classically, maybe instead of giving you the sequence of individual species and asking you to build the tree, instead the data you have is, all the species from North America or South America or Europe or Asia mixed together, and I'm asking you to build the tree in that way. And can you still do it? It's actually, quite a challenging and interesting problem of whether or not you could do this, and it's one that's been worked on extensively over the past 5 or so years. So can you take this frequency matrix, this sort of analog data, and can you sort of unmix it, if you will, where you have to build not only the tree, but you have to uncover what the leaves are, what the tree that relates those leaves are, and also what the proportions of each leaf are in each sample. Okay? So it's quite a challenging problem. There's a lot of algorithms that have been developed by a number of the people in this room actually to solve this problem. And I'm not going to go into any more detail about that now because I wanted to sort of bring it back to using some of these algorithms to actually understand metastases. So there's 2 parts to the talk. In the first part, we're going to talk about metastases, assuming we've built this tree, and we're not going to worry about any of the issues involved in building this tree. And in the second part of the talk, if there's time, I'll actually delve into a little more of the details of some more recent work on copy number aberrations and how we can deal with some of the complexities there. Okay, so I guess as way of introduction, so now I showed you some work where they use standard techniques and tried to build these trees, and they got these weird homoplasia events. In other lines of more recent work, the analysis is more sophisticated, using these techniques that build clone trees. Then from these clone trees, the researchers attempted to reconstruct this history of migrations between sites. And in doing so, some really interesting phenomena were observed. So the contrast isn't great here, but this is a paper from a few years ago showing sequencing of several patients. So each of these panels here, this is actually one patient. Part B, there's 3 different patients from with metastatic prostate cancer. And in each patient, the primary tumor and multiple metastases from the same patient were sequenced. And what was found after building these clone trees and then analyzing what the migrations between the different metastases must have been was that these patterns were extremely complex. And this idea of monoclonal seeding of metastasis, where each metastasis was founded by only a single migration event, seemed not to hold at all in this data. So you can see here in the primary tumor in the prostate, there's sort of this first wave of migration seeding these local metastases, then a second wave of migration seeding more distant metastases, and somehow then just cells migrating all over the place, back and forth between different metastases, coming back where they've been before, multiple cells going to different locations. Here's another patient, again, just each of these arrows, and you can't really see the heads and tails of the arrows, unfortunately, but just migration back and forth all between these different sites. And so the conclusion was, 5 of, I think, the 10 or 11 patients they analyzed in this data set had what they called polyclonal seeding of metastases. Or even reseeding, where cells came back to sites where they were before. Another example from a similar type of study where in a single patient, primary tumor and multiple metastases were sequenced. This one in breast cancer, the sort of illustration is a little bit different. Here the primary tumor at the bottom and the sort of thick branches are kind of showing you the clonal structure of the tumor. So you think of these as populations of cells. So here this purple branch is a new clone that sort of grows out in the population, another new clone, and here's the different anatomical sites. And so if you sort of look at this picture and think for a little bit about what's going on here, you see that in the kidney met, there are multiple clones that are somehow present in the kidney met, multiple clones that are present in the liver met, but multiple clones are shared between these 2 mets. You can see this by the shared gray and orange and green. So again, somehow this metastasis was seeded by not one, but many clones migrating. And that was sort of the conclusion of this paper. They had what they called, in this case, multi-clonal seeding. So these studies are actually quite interesting and really, contradicting the dominant paradigm of monoclonal seeding and metastasis has been sort of the paradigm for decades. And so this has led to a lot of discussion about, how much do we really understand about the process of metastasis? Is a metastasis a monoclonal seeding event? Is it seeded by multiple cells, polyclonal? Is metastasis only a result of migration from the primary tumor, or can you have cells migrating between different metastases? Can cells return to sites where they've previously been, the so-called reseeding or self-seeding? And there's a lot of discussion about this, and really at the time when we started working on this, there wasn't really what we thought a very rigorous approach to answer these questions from DNA sequencing data. And that's what we wanted to try to attempt. So the first issue, as I discussed, was this homogeneity issue. Okay, so we sort of addressed that issue by building these clone trees, or maybe we can really address this issue by doing single cell sequencing data, so that each leaf of our tree is a single cell, okay? But even if we construct this clone tree, or we do single cell sequencing perfectly with no error, if we have the perfect clone tree, the perfect representative of the mutational history of this tumor, that actually is still not enough to tell you what the history of migrations were. And that's because there are really two processes going on in this picture. One process is cell division and mutation is a marker for cell division, and this tree describes the history of cell division if we've done it perfectly. But the other process is migration. And so one way we could represent migration sort of is with this migration graph, where each node is an anatomical site, and we have a directed edge indicating a migration of one cell between sites. So this picture that I've shown you here, here's the reconstruction of the cell division and mutational history of this tumor, and here's the picture of the migration history of this tumor. And already you can see that these two processes are somehow correlated, but they're not the same. In particular, this structure right here is not a tree. So what exactly is the relationship between these two processes, and how do we sort of link them together? Right? Now remember, I've abstracted away, this tree is perfect. There's no error in this clone tree. Yet still something is missing, something that allows me to infer the migration history. And what's missing is that even if we've constructed this tree perfectly, we only know information about the anatomical site at the present day. So we only know the anatomical site on the leaves. And we don't know the anatomical site of any of the ancestors because we didn't measure them. And without that information, I can't tell you, in fact, anything about the history of cell division, or history of migrations in this tumor. Okay, so how can we recover this information? Well, it's an evolutionary analysis. We didn't directly measure the anatomical site of any of the ancestors, so we have to infer it. We have to guess. So the way we would guess is we have to label each of these ancestral nodes by an anatomical site. So here I've used these colors to indicate the three sites, so it's really just, coloring each of the ancestral nodes of the tree. And so if I color in this way, I've just guessed that, the root was the primary, and, here's the different metastases where these cells were at, MET1 or MET2, then I can take this label tree, and now I can, from this label tree, describe, because each time there's a color change that indicates a migration, so I can add one of these directed edges. And in total, if I look at this labeling of the tree, I get 8 migrations. And say, okay, there it is. There's a reconstruction of the migration history of this tumor, and it took a total of 8 migrations to do this. But that was just a guess. Could we do better? Could we do something different? Of course, we can label this anyway. So if I instead try to guess that each of the ancestral nodes was at the site of the primary tumor, then the migrations only happen on these edges right here, and we get a total of 4 migrations. Well, now we can start to think about how we might evaluate these different migration histories. And if we think that a cell leaving one anatomical site, traveling through the bloodstream, and seeding a tumor at another anatomical site is a pretty rare event, then one thing we might like to do is minimize the number of such rare events. And that's, in phylogenetic terms, maximum parsimony. So we could actually just try to take a clone tree and label each ancestral vertex by a color or anatomical site to minimize the number of migrations. And this is just a classic phylogenetic problem. This is just classic single character maximum parsimony, and you can solve this problem very efficiently. And in fact, this construction has been used to infer migrations in other contexts, in this paper in the 80s from Slatkin and Madison. So we can do this, and in fact, Saurabh Shah, who's not here yet, but soon will be, in a very beautiful paper, actually did use this type of approach where they built a clone tree and then they used maximum parsimony to label the ancestral vertices of this clone tree in order to minimize the number of migrations. And they did this on several patients with ovarian cancer. Here's one such patient where there was tumor in either the left or the right ovary, which had spread and seeded new tumors throughout the abdominal cavity. And so they built a clone tree relating these tumors. They had the leaves, labeled according to the anatomical site where they were measured, and then they inferred the anatomical site of the ancestral node. And if you take their representation that was reconstructed and now show it in this migration graph form, what you get is this picture where there was a total of 13 migrations and a very complicated migration pattern. So here the primary tumor is the right ovary, multiple cells migrating from the right ovary to the small bowel, from the small bowel seeding tumors at other anatomical sites, the fallopian tube, the appendix, etc. If you see here, look, the left ovary somehow receives cells from the small bowel and multiple cells from the left ovary, 13 migrations, very complicated. Yes, Quaid? Sorry? Yeah, leaf labels are provided because you know where the site of every cell you measured. I mean, you sequence the left ovary and you measure the mutations there. You sequence the right ovary, you measure the mutations there. You know where each of the clones were present because you sequence them there. Well, then you still will know that it was the same clone and you measured it in two different sites. If it was single cell sequencing, you definitely know this information. With clone tree, there's, yeah. Yeah, you could, and in fact, that's, I don't know if that's done in this picture, but yes, you could. You could have multiple, different clones at different anatomical sites, and it does happen. Okay. There's no clock in here in this church, like priests just get to give a sermon as long as they want, right? All right. Move along here. All right, so this is it. So 13 migrations, and that's the minimum, yeah. Ah, that's a great question, and that's the next slide. So this is the minimum number. This is the minimum number. Yeah, maybe you guys are just going to tell me the next slide. Yeah. The same clone, same mutations, but in two different sites. It's just represented in the tree twice. It's two leaves that are present in different. Oh, an ancestral clone present in two different sites. That's a good question. So here we have to choose one or the other labeling, and that will cause actually sort of this extra counting of migrations. It turns out to be actually an important issue. For single, if you're at single cell resolution, this would never happen, right? Because we think the cell with the actual same identical mutations would not be present in two different sites. But there is some loss of resolution when you go to clones, and it could happen. Yeah. Well, the mutation content of the ancestral clones is inferred, and we're assuming we did that perfectly. So we've got the mutation history of the tumor perfectly. You're going to overcount. Yeah. Yeah, exactly. Yeah, sure. Yeah. But you guys are worried about actually a much more complex issue. There's a much simpler issue that I think the first question was concerned about. These are like second or third order issues. the most important issue, even for this simple model, is that the minimum number of migrations is not necessarily a unique tree, a unique labeling. And even in this simple data, here are 5 different migration graphs. There are 5 migration graphs that all have the same minimum number of 13 migrations. And now the pattern is quite different. So remember the small bowel is sort of an intermediate here. Now there's sort of cells going back and forth between the small bowel. Look, actually, even the site of the primary has changed between these tumors. Here, the small bowel is not an intermediate. You know, everything comes from the left ovary. So in fact, perfect clone tree, multiple labelings that give the same minimum number of migrations, the migration history quite different. Right? So how could we attempt to resolve this issue? Well, we need some other criteria by which to try to distinguish these trees. Is there some way we could attempt to, score these trees differently? So one idea would be, well, we might know some information about the probability or the weight, if we're in a parsimony setting, of a migration happening between 2 sites. And maybe we'd say it's sort of much more likely that you migrate to a close anatomical site than to a distant one. And so we could certainly try to incorporate that information. Unfortunately, we don't really have any good way of parameterizing such a model at this point, because we don't really have these good probabilities for migrations between different anatomical sites. So that is perhaps not as helpful at the moment as one would hope. But what we could also try to do is score different migration graphs, and we could try to develop some way of distinguishing different structures of the migration graph. And there's 2 structures I want to sort of point to and suggest might be interesting. One is the existence of parallel edges, and the other is just the topology of the migration graph itself. So these parallel edges, where you have multiple migrations between the same pair of anatomical sites, is interesting actually for 2 reasons. One is that there's some evidence that tumor cells migrate through the bloodstream not as single cells, but as clusters. Groups of maybe 3 to 5 or perhaps more cells that are migrating together. And if those cells had different mutations, then they would be a parallel event. And so that would indicate these parallel edges in the migration graph. Alternatively, these migrations may not be happening in clusters, but it could be that parallel edges are preferred because at a certain time in the evolution of the tumor, there might be a primary, a tumor that's able to kick out cells, and there might be an anatomical site that's sort of receptive and able to receive cells. So maybe even if they're not happening as clusters, they could be happening contemporaneously. And so we can now think about perhaps scoring these graphs differently if we count not the number of edges in this graph, but the number of groups of edges. So we could count parallel edges as a single event, a so-called co-migration event. And now, by counting co-migrations, we can start to distinguish some of these topologies of the tree. And then the other thing we can do is just look itself at the topology of the migration graph. And in fact, there's really just 6 such graphs in terms of their structure. First, we can define whether the graph has parallel edges or not. These are so-called monoclonal migrations or polyclonal migrations. And then we can distinguish whether the graph is a tree, or whether it has directed cycles, or whether it has undirected cycles. Okay? And those 6 different taxonomies give us 6 different ways of describing the migration pattern between the trees. So the simplest, a tree with single edges, is our monoclonal single source seeding. The most complicated, when we have parallel edges and we have cycles, directed cycles, is polyclonal reseeding. Okay, we've got names for each of these, yeah. Yeah, the way we sort of describe this is that if you've got at least one parallel edge, we call it polyclonal, at least one. Yeah, and then if you have, at least one, if you're a DAG, then you're directed. If you've got at least one directed cycle, we call it reseeding. So if you do this kind of, at least one, yes, but so we sort of describe the whole graph this way. All right, so now we can attempt to introduce these 2 scores. And if we introduce these 2 extra scores, now we've got 3 knobs we can turn as we try to label these ancestral nodes. We can try to minimize the migration number. We could try to minimize this co-migration number by counting parallel edges, or we could try to, examine what happens in terms of the number of migrations if we allow different migration patterns. So if we don't restrict this at all, then we might, require a certain number of migrations. If we want to ask, well, I don't think that reseeding happens, so let me rule out reseeding. Then how many migrations and co-migrations could I obtain? And now this simple problem of find a labeling that minimizes the number of migrations becomes a much more complicated problem. It's actually a multi-objective problem in that as you try to look at trade-offs between these different objectives, you get different optimal solutions. So there's a trade-off between migrations, co-migrations, and allowed migration patterns. And so now you can sort of pose this as what we call the parsimonious migration history problem. Given the clone tree, try to label the ancestral nodes to minimize the number of migrations and co-migrations for a given set of allowed migration patterns. Okay, so if we allow any pattern, and we don't worry about counting migrations, then that's our usual maximum parsimony problem. That one we can solve efficiently. Once we also try to minimize co-migrations, then it becomes a more challenging problem, and we just solve it via an integer linear program in a way that I won't describe. So here, we see this example where we've labeled, and the minimum number is 4 migrations and 2 co-migrations. And this is what we would call a polyclonal, because there's parallel edges, but single source seeding, because the underlying tree, the underlying graph is a tree. So now if we do this on the ovarian data, and this is the trade-off between the two objectives, here migration number, here co-migration number, we see that we can start to distinguish these different migration patterns. So here was the published one where there were 13 migrations and 10 co-migrations, but we could do better. We can actually get 7 co-migrations, so this is perhaps a more parsimonious tree in terms of co-migrations, and this one has reseeding. Here's another one that also is 13 and 7 in reseeding. And then if we say, well, I don't believe in reseeding, I don't believe that cells could come back to where they were before, is there a pattern where there's only single source migrations? And if we add that restriction, then we can find this labeling, and this labeling is single source, polyclonal, and it takes 14 migrations. So now you could ask, well, what do you think is more likely, that it was only 13 migrations, but a very complicated pattern, or that there was one more migration, and the migration pattern was much simpler? It was a tree with no reseeding, no things going back and forth. Now, we can't answer this question for this data, unfortunately, because it's just one data set, and it's just different possibilities. But now, if you start to do this across different data sets, you could start to look at the trade-offs between these different criteria, and perhaps start to examine in a more rigorous way, and even bring this in a more probabilistic way, and look at the trade-offs in these different criteria. Okay, so that was assuming we had the clone tree. We label the ancestral vertices to find a migration graph that's parsimonious, but we could also actually do this in reverse. We could actually use information about what we think the migration graph might be in order to help us resolve ambiguities in the clone tree. And we could have many ambiguities, but one of the most common in doing this is that you might have a tree that has a polytomy or a multifurcation where there's a node with a parent with more than 2 children. And if this was, of course, a single cell tree, we know that a cell divides into exactly 2 daughter cells. And so this is some loss of unresolved ancestral relationship in the tree. And now what we can try to do is resolve this ambiguity using information about the migration patterns. Okay, so if we took this tree and we believe this tree, we would get this pattern that I showed you here. If instead we sort of relax the criteria for this tree, and in fact I sort of showed you these varying allele frequencies, there's always some ambiguity in how you construct these trees, and so we can sort of look at this ambiguity and look at variability in the varying allele frequencies, and combine this with the migration inference, and do both at the same time, where we try to find a parsimonious migration history and build the clone tree at the same time. And if we do that in this simple case, we might resolve this polytomy in a way that gives us a simpler migration history here, the monoclonal single source seeding. Okay, so this is sort of a nice algorithmic problem. Does it really have any bearing on real data? So if we go back and look at this breast cancer data where there was this very complicated multiclonal pattern of migrations, we can sort of represent this type of tree in the migration graph format. Here's the primary tumor, the breast, which sends cells to the rib and the lung, and the rib sees all these other multiple migrations to these other sites. A total of 14 migrations, a very complicated migration pattern. If we look at the ambiguities in the construction of this tree and account for those ambiguities, we can actually obtain a much simpler reconstruction where we get only 5 migrations, and it's the monoclonal pattern, the sort of standard thing we're used to, where from the breast we have a single migration to the lung, followed by, from the lung, multiple migrations to the tumor. So this sort of shows you, that all the issues that can arise. And so if you're gonna sort of reject the monoclonal seeding pattern, you really want to have strong evidence for it. And so in this particular example, we would argue that the evidence is not so strong for a polyclonal pattern, because with just slight adjustments in these variant allele frequencies, allowing for slight errors, you get actually the very simple monoclonal pattern. So the conclusion here is that these complex migration patterns really require strong evidence. The null hypothesis, if you will, is that migration is a very simple event, monoclonal. To reject that null, you really want to have strong evidence. In order to really address whether you have strong evidence, you need a way to kind of look at these different migration patterns and to score them. And so this algorithm, it's got this name Machina, allows one to explore these trade-offs in the migration patterns, and then also to simultaneously look at the clone tree and the ancestral and the migration history. And of course, all of this is to some extent to help us really get a notion of what exactly were the migrating clones. And in order to really understand what the drivers of metastases are, we need to not just have matched primary and metastasis samples. We'd like to really know what exactly mutations were present in the clone that migrated. So the better we can do these ancestral reconstructions as we look at these across patients, this will give us higher accuracy in sort of doing these studies where we try to look for the drivers of metastases. So that was the summary of the first part. Oh my gosh, and because we started late and we're done, maybe this is the end, huh? Quaid would be very disappointed. Maybe I should just take questions here. So I'll just tell you the second part. What can I tell you about the second part quickly? Maybe I shouldn't even try. I'll take the question here. So, sorry, the same? Well, no, not for this necessarily. I mean, here, build the clone tree or build the cell tree, build it perfectly. It could have homoplasy, it could have whatever it has. It describes the cell division history perfectly. And you're still going to have this ambiguity where you can't necessarily describe the migration history. So it's not about mutations occurring, twice or not. That's all represented in the cell division history of the tumor that you've inferred from the mutations. It's possible. I mean, I think if potentially treatment could exert some strong selective pressure on the tumor and maybe lead to higher rates of homoplasy than you might otherwise assume. So that could be perhaps one of the issues that arise when you kind of do these analyses without being aware of all the issues of mixtures, et cetera. And it is true that many of the methods that attempt to build clone trees from bulk data, which is a challenging problem that I sort of skipped, many of those methods do make this no homoplasy assumption, because it's very hard to solve this, build a tree from mixtures of the leaves, and in order to make that problem tractable, often people will make the no homoplasy assumption. So that's true, but in terms of migration histories, in everything I said here, it doesn't matter. You're given the true clone tree. Sure, yeah. Yeah, no, and that's a great suggestion. That's sort of one of the, one of the ideas for, we'd love to, it's probably, we do know that there are biases in the migration patterns. I mean, you can go look at just clinical data and see that, certain tumors have preferential sites by which they migrate to, and one can make use of that data. There isn't really enough of it at the level of resolution that we would need for some of the analyses I showed. I mean, for ovarian tumors, we don't really know the probability that it would, cells would migrate to the small bowel versus the, left fallopian tube. There isn't that level of information in the clinical records, but if, you start looking at enough of these trees and you start looking at the trade-offs between the different migration patterns, then you can attempt to, learn the parameters of the biases in the migration patterns. Yeah, so that's actually one of the things we, like to do going forward. I mean, well, so, yeah, so copy number is the big killer here. Since you asked the question, I can quickly say two things. So there's tons of copy number, copy number everywhere. Copy number makes this whole problem really hard because if you try to build these trees from mixed samples and you allow for copy number aberrations, you just get huge amounts of ambiguity, even if you make the no homoplazy assumption in there. And curiously, and I'm not going to say much more, because I won't jump in. What's that? It's only the tree construction, yeah. So again, everything I said about migrations, you can assume you have the true cell division history that you've reconstructed from copy number, single nucleotide mutations, structural variants, whatever you want. You've got the true cell division history, and you still have this ambiguity in the migrations. But one thing we have been interested in is, curiously, in many of the data sets, wait, let me just, I'm not going to show you all this stuff. You have to skip all this stuff. The reason we went back to this sort of classic problem of how you infer copy number states or mixtures is because, sort of curiously, a lot of the data where there was polyclonal migrations reported were in tumors like the prostate tumor where there was extensive copy number aberrations. In ovarian tumors with this extensive copy number aberrations. The breast tumor example, also extensive copy number aberrations. And so we were really worried about how reliable are those clone trees because, of course, it's very hard to get the clone tree when there's copy number aberrations. So we went back to this copy number problem. And then on the flip side, some of the studies like in pancreatic cancer where there are actually also extensive copy number aberrations and whole genome duplications, they were reporting very simple migration patterns, monoclonal migrations, but the copy number was not, they were just assumed all these cells were diploid, which is very likely not to be the case. So there's this sort of curious thing where, you got complex migration patterns in tumors with copy number aberrations where getting the clone tree is really hard to do. And then, in cases where you ignore all the copy number aberrations, even though they very likely exist, you get very simple migration patterns. So, if you really want to answer whether or not metastasis is polyclonal or monoclonal, you really need to know the clone tree reliably, and it's really hard to do it in the context of copy number aberration. So we've gone back to this problem and tried to do a better job at doing it, but that'll be the topic for another talk.